In lecture video C, we are now going to talk about translation in detail. We will cover slides 15 through 22. So one of the first things that we said was that we're now looking at different types of RNA. So when we talk about translation, this process of taking messenger RNA and stringing together the correct sequence of amino acids to make our protein, well, this is where another kind of RNA becomes critical. So here we're going to begin talking about transfer RNA, or tRNA. Recall that when we looked at that genetic code, we said that there was one special codon. It was in green. It was AUG, and it was our start codon. So translation is going to begin at the messenger RNA where AUG is present. That AUG start codon also codes for an amino acid. This is the only one that you have to know. Do you remember what it is? AUG start codon also codes for methionine. So this is going to be important in how we set our reading frame of our codons. Remember, 3 mRNA nucleotides at a time. Well, why is it 3 RNA nucleotides? So those 3 messenger RNA nucleotides that make up a codon correspond with 3 tRNA nucleotides. We call these 3 tRNA nucleotides the anticodon. And these are going to pair according to base pairing rules. So that is what is depicted down here, okay? We have our blue transfer RNA example here. So this line represents our RNA nucleotides. There is a special part down here where we have 3 codons that are our anticodon. Our anticodon is complementary to our codon. Okay, so this tRNA anticodon is going to have base pairing with the mRNA codon. So where we have AUG as our start codon, the anticodon that's going to allow for this tRNA to come in with methionine as its amino acid up here is going to be UAC. It's base pairing. Where we have an A, we will have a U. Vice versa, where we have a U, we will have an A. C and G are going to pair here. So this anticodon is what's going to be important in bringing in the correct amino acid. It's that anticodon on the tRNA that is going to match the codon on the mRNA as our ribosome is translating that messenger RNA into a protein. Okay, we said that for translation, we're going to have initiation, elongation, and termination. What we are elongating here is our protein. So when we look at our ribosomes, ribosomes are going to have 2 subunits, a small subunit. In bacteria, it's the 30S subunit, and a big subunit. In bacteria, it's 50S. Remember, our cells also have 2 subunits, but they're different kinds. They're different versions of those subunits. Okay, so for initiation, what are we looking for? Well, we're looking for that start codon on the messenger RNA. And so we have these 3 little pockets that we see on our 30S small subunit. Well, AUG is going to sit right in here. That AUG codon is going to correspond with the UAC anticodon. This tRNA is going to have methionine as its amino acid attached to it. So once we get this setup done, then we're going to have our larger 50S subunit come in. it has these nice 3 little pockets. So our large subunit and our small subunit know how to fit together, essentially, kind of like puzzle pieces. So the well, let's start over here. Let's actually start with A. The A site, I like to think of it as the acceptor site. So this is where our incoming amino acid is going to be. P is where we're going to have our protein elongating from. And then E is kind of the exit or the ejector site. If that helps you remember the E, P, and A site, go for it. That's kind of an easy way to remember what's going on at these 3 different sites. So initiation is all about getting everything set up and aligned so that we are reading the mRNA sequence in the correct codon pattern. AUG sets the reading frame. All right, so then we have our next amino acid brought in according to the codon that is present on the messenger RNA. Well, we're looking for the matching anti-codon. Only the tRNAs with the matching anti-codons can come in. What we see is a peptide bond forming between our 2 amino acids. And then what we're going to do is we're going to shift that whole thing down. And we're going to have our empty tRNA eventually ejected or exited from that E site. So we do this. We read it codon at a time, bringing in the correct tRNA with the corresponding anti-codon. And then we shift down after we form that covalent bond. So much kind of like, I don't know, like we clamp down and we read 1 nucleotide at a time, one codon, the next codon, the next codon. And that ribosome is going to move down the length of that messenger RNA. And we're going to keep going and going and going and going until we reach one of those 3 stop codons. UAA, UAG, and UGA are anti-codons on tRNAs that do not have an amino acid attached to them. So what happens is that when those guys come in that A site, well, ribosome goes, oh, there's nothing here to do. And eventually we'll just eject that free transfer RNA that we don't need anymore. We're going to have our ribosome, those 2 subunits will come apart, and our messenger RNA may either be read again, or if we're done utilizing that messenger RNA to produce our proteins, then we may go ahead and degrade that messenger RNA. As we are making our long chain of amino acids, recall that those R groups, those side chains, are going to cause our protein to fold up into its correct 3-dimensional shape. And it will perform whatever function it's supposed to perform. Maybe that protein will go and be embedded in the cell membrane or the plasma membrane. Remember, those 2 are synonymous. It might be a structural protein, or maybe this protein is an enzyme and it will have an active site where it can catalyze a given reaction. So when we start looking at this, you should make sure that you understand how DNA, that template strand of DNA, is transcribed into messenger RNA using RNA polymerase, and how our ribosomes read that messenger RNA 3 nucleotides at a time, and how those 3 nucleotides correspond, that codon, how it corresponds with a given amino acid. It's all about the pairing of that codon with its anti-codon. So I'm going to show you again an animation from your textbook so you can see this in action. There is far more information here than I expect you to know. I don't care about initiation factors or the energy usage on this one, but I want you to see this process in action.
Protein synthesis occurs in 3 phases: initiation, elongation, and termination. In bacteria, such as E. coli, initiation requires the association of several components: small proteins called initiation factors, the first tRNA, the mRNA to be translated, and the small subunit of the ribosome, also called the 30S subunit. The key event of initiation is the association of the mRNA and 30S subunit so that the start codon, AUG, is centered on the ribosomal subunit. The 50S subunit docks to complete the initiation complex. Initiation uses energy by hydrolysis of GTP. Note that the ribosome has 3 tRNA binding sites: an acceptor, A site, also called amino acyl site, a peptide, P site, and an exit, E site. The ribosome is ready for elongating a chain of amino acids. Elongation requires an elongation factor called EFTU, associated with GTP, to bind to amino acyl tRNAs. This complex enters the A site. Correct selection of the tRNA complex depends on codon-anticodon pairing. Here the anticodon CCC pairs with a GGG codon. GGG codes for the amino acid glycine, which this tRNA carries. Next, the ribosome catalyzes the formation of a peptide bond between the 2 amino acids, and the hydrolysis of GTP drives peptide bond formation. The resulting EFTU, GDP, is expelled. An elongation factor called EFG, associated with GTP, binds to the ribosome. The GTP is hydrolyzed, providing the energy to ratchet the 50S and 30S subunits ahead 1 codon. This maneuver opens up the A site, and the uncharged tRNA slides into the E site. The next amino acyl tRNA that enters the A site creates a conformational change in the ribosome that telegraphs through to the E site and ejects the uncharged tRNA. These elongation steps repeat along the mRNA.
This is what I want you to see, this movement. We ratchet forward.
Eventually the ribosome arrives at the end of the coding region, marked by one of 3 possible stop codons. This phase of translation is called termination. No corresponding tRNAs exist for stop codons. Instead, a protein called a release factor, which has the general shape of a tRNA, enters the A site. The release factor triggers hydrolysis of the peptide tRNA bond, thus releasing the peptide. The 30S and 50S subunits fall off the mRNA and separate in the cytoplasm, available once again to form a new translation complex with another mRNA.
Like I said, more there than I expect you to know, but I want you to see that action, that movement as we slide down our messenger RNA. All right, so I want to jump back and go more into detail about how bacteria uniquely and very efficiently regulate their gene expression. So while I don't care that you understand and know the details of energy usage in these processes, realize that making proteins uses energy within the cells. And so bacterial cells don't want to make proteins if they don't need them. So the cells have the ability to turn on and turn off the use of their genes. So recall that the promoter part of a gene is the beginning part. It's essentially the beginning part of the gene. It's the DNA sequence that our RNA polymerase is going to look for. Well, if we block off this promoter region, it essentially serves as a way for us to turn off the gene that follows the promoter. Because if RNA polymerase can't get to the promoter, well, it's not going to make messenger RNA and we won't make the protein. So we have these regulatory processes in our cells too, but one of the things that is unique to bacteria is that they can have a single promoter region that controls multiple genes downstream. Those genes that are all controlled by that single promoter oftentimes are going to be for one particular process, like metabolizing a given nutrient. Well, if that nutrient is available, we want to turn that whole operon on. We want to make all of the proteins needed to metabolize those nutrients. But if those nutrients aren't there, we want to be able to shut that whole thing down. So when we have an operon, this promoter that controls multiple genes, what's going to happen is that when it is induced, when it's turned on, we will make one single messenger RNA from all of those genes, and then we will transcribe those proteins as we needed. What this system does is it really allows bacteria to be energy efficient. If they don't need certain gene products, they're not going to express those genes. So they'll have them turned off. We say that they are repressed. Okay, so induction means that genes are turned on. We are actively transcribing, translating, we are making those proteins. But if we don't have a need to induce those genes and make those proteins, bacterial cells want to repress them. So they will turn all of them off. They won't be transcribed, we won't be translated, and no protein will be made. If we don't need those proteins, we're not going to waste that energy. So one example of an operon is the lactose operon in E. coli. We know a ton about this particular operon. We shorten its name to the lac operon. I'm not interested in you understanding a whole lot of detail about the naming system here, but what I want to point out to you is that this is our promoter region. We have multiple genes that are controlled by that promoter region. So we've got an enzyme here, beta-galactosidase. We have lactose permease. We have a couple of other enzymes. Those enzymes those genes code for enzymes, and those enzymes are responsible for metabolizing lactose. Well, if lactose is available, do you want those genes induced or repressed? If lactose is available, you want to induce those genes, turn them on, make the enzymes to break down lactose, and use it. If lactose is not available, what do you want to do? Well, if you're a cell, to save energy, you want to repress those genes. So with this operon, all of those genes are controlled by that one promoter. So there is a structure that is called a repressor. This repressor molecule physically attaches to the promoter. By having that repressor attached to the promoter region, it blocks that RNA polymerase holoenzyme from getting to it. And it prevents our enzymes from being made. So when we have that repressor attached to our promoter, the whole operon is repressed. In the presence of lactose, lactose will cause a conformational change, and the repressor will fall off, and then we will have that promoter available to our RNA polymerase holoenzyme. It will transcribe all of those genes in one single messenger RNA. Our ribosomes will translate those individual protein enzymes, and then those enzymes can go to work breaking down our lactose, and we can actually use it. Now, when lactose when we've used all of the lactose and there's no more in the environment, what do you think will happen within the bacterial cell? That repressor is going to come back on and it's going to bind to the promoter, and we will go back to that repressed state. And then when we have lactose, we will induce the operon. When we don't have lactose, we will repress it. Okay, so let's watch a video of this.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon: beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
This is a beautifully elegant way of making only what it is that you need. So this is an incredibly energy efficient way for bacteria to utilize the information within their genes. All right, we will wrap up Chapter 8. There is your reading assignment, and here are the objective review and review questions for you to look over.